Tuberculosis is one of the most deadly infectious diseases on the planet and is known to be caused by Mycobacterium tuberculosis. In this video, we will investigate the process of primary infection. Shown here is the surface of the MTB in red. There are various antigens embedded within a thick layer of various lipids and mycolic acid. This layer, which is distinctive to Mycobacterium, acts as a shield to protect it from host defense systems and helping the manipulation of macrophages. MTB thrives inside the host body by entering macrophages because its internal environment is more beneficial to MTB survival than the extracellular space. Here we can see one of the most important ligand and receptor interactions. Lipoarabinomannan, or MANLAM, is a glycolipid ligand expressed on the surface of MTB. MANLAM interacts with the macrophage mannose receptor. We will now look at this at the nano level. The mannose receptor consists of multiple extracellular domains which allow the recognition of a diverse range of pathogenic ligands. Here we can see the 5 most important carbohydrate binding domains, or lectin domains, of the mannose receptor, which are known to recognize and bind the monosaccharide sugars called mannose groups that cap the MANLAM ligand. A promising therapy to reduce MTB's virulence would be to interfere with the interaction between MANLAM and the mannose receptor. Once MANLAM is recognized by the mannose receptor, the MTB gets engulfed by the macrophage and resides inside a phagosome. One important mechanism for MTB survival in the phagosome is the prevention of maturation into an endosome. Infected phagosomes convert Rab5 to Rab7, and this affects host signaling and prevents maturation. Rab5 is a protein necessary for early endosome formation. Another mechanism of MTB survival is the inhibition of phagosome-lysosome fusion and inhibition of phagosomal acidification. Without fusion, the phagosomal lumen remains at only a mildly acidic pH and MTB is not degraded. Now let's explore this at the nano level. This is the protein VATPase, which is bound to the membrane of the phagosome. In the phagosome-lysosome fusion process, subunit H of the VATPase binds to the protein VPS33B, which is part of the Hobbs complex and is attached to the lysosome. This induces phagosome-lysosome fusion, leading to phagosomal acidification. However, in the presence of MTB in the active phase, a protein called PTPA is secreted by MTB into the cytosol, and PTPA is able to bind to subunit H. This physically blocks the binding site and therefore inhibits the binding of VPS33B. Furthermore, PTPA dephosphorylates VPS33B, which then stops the maturation of the phagosome and therefore inhibits the acidification process. The success of Mycobacterium tuberculosis rests upon its ability to convert macrophages into a permissive cellular niche. As we've shown, the complex series of measures and countermeasures between host and pathogen ultimately determines the outcome of primary infection. Thank you for listening.